My dear students, you must have visited your nearby aquarium shop and explored the colorful world of fishes inside water. Now, if I say they are not just pets, they are examples of genetically modified animals. Yes, with the help of biotechnology, we have converted this normal tetrafish into glowfish. Now, to know how, you have to watch the one-shot video of the chapter Biotechnology and its application in our channel, that is PW NEET English, where we learn with fun. And you are with Biswajit, your zoology master. We'll complete the chapter, we'll learn the concept, and we'll practice questions. For that, you just have to click the link given here, and that will lead you towards the new world, that is PW app. And there is something new for you, that is PW NEET English WhatsApp channel. You just have to scan the QR and that will redirect you into the channel. And there you can get a lot more, starting from DPPs, polls, personalized strategies, questions, diagrams, mind maps, and many more. So what are you waiting for? Let's jump in. So what are the topics we're gonna cover for this particular chapter? The topics will be obviously the applications of biotechnology. We'll talk about different aspects of application or applied biotechnology, the application of biotechnology into agriculture, application of biotechnology into health science or medicine, application of biotechnology to form genetically modified organisms or transgenic animals. We'll talk about some ethical issues related to the application-based biotechnology. Okay. So, basically, we have learned into the previous chapter, that was biotechnology principles and processes, how to construct recombinant DNA, the method of altering genetic makeup of DNA and RNA, and to introduce that genetically modified or recombinant DNA into a new organism or host organism. Okay. So biotechnology has many applications such as biopharmaceuticals, therapeutics, diagnostics, genetically modified crops. We'll talk about BT cotton, BT brinjal, BT tomato, processed food, bioremediation, waste treatment, and energy production. Yes. And the three critical research areas of biotechnology is given here. First, aimed towards providing best catalyst. Now, what do you mean by catalyst? The enzymes. More the efficiency of enzymes will give us more purified or more efficient product. Ultimately, the biosynthetic mechanism or the biological pathways always undergoing inside our cells with the help of these enzymes or biocatalysts. Okay. Second thing, creating optimal conditions. Now, inside your laboratory or inside those bioreactors, already we have studied into the previous chapter, if you fail to provide the optimal condition, that will lead to nowhere. That's why with the help of biotechnology, that is the fusion of biological science with modern technology, that will lead us towards the best laboratory practices. And third thing, the downstream processing technologies. Now you know downstream processing means it's DSP, separation and purification. Now you have obtained the product from the genetically modified organisms. You have utilized the best technologies inside your bioreactors. Now it's time to extract those products. Either they can be antibiotics or some drugs or some enzymes or some other proteins. Now until and unless you are separating them from the impurities and purifying them before, just prior to market them, right? You have a market to sell those products. So the best downstream processing technologies will help us to produce the best product. Now, one of the example I am giving you here, suppose for fever, we take the medicine paracetamol, right? So paracetamol is available in the market in different trade names. Few are selling by the name of Calpol, few are selling by the name of Dolo, few are selling by the name of Paracep, right? Now for different brands, you will find the price is different. Like for a tablet, for some brand they're charging 5 rupees per tablet. For other brand they're charging 3 rupees per tablet. For any other brand, third brand, they are charging 2 rupees per paracetamol tablet. And suppose the concentration of paracetamol is 650 milligram inside every tablet, then why different companies, different brands are charging different charges? Basically, the brand which is charging 5 rupees quite like costly, right? So basically they are utilizing good condition, optimal condition while they are preparing that drug. They are providing best downstream processing technology while they are separating and purifying that drug from that mixture, from that bioreactor. So it's based upon the technology you are using that will decide the market price or the quality of your product, right? Okay. So basically, in which all sectors we can utilize biotechnology. Obviously, the prime sector is agriculture. We know that the primary utility of biotechnology is inside food science, and agriculture deals with food science. We have to talk about crop production, genetically modified crops. We'll talk about different transgenic varieties of animals. Obviously, we utilize biotechnology and its application in industrial sector. When you talk about sustainable development, we'll talk about biotechnology and application in medicine, right? We'll talk about biotechnology in environmental science. That's called environmental biotechnology. The terminologies we are using: bioremediation, sewage treatment, biodegradable plastids, waste management, modern day aspects of all these things. When you talk about transgenic animals, you might have heard about the animal called liger. Yes, this is a picture of a liger. Basically, that is a hybrid of lion and tiger. Now, with the help of biotechnology, we were able to produce this kind of hybrid animals or transgenic animals. And obviously, the last thing we'll talk about the ethical and social issues. Many kind of legal terms will be used: IPR, intellectual property rights, biopiracy. We'll learn one by one. Okay, so first thing we'll start with application of biotechnology in agriculture. Okay, so basically the agriculture started booming from the time of Green Revolution. Okay, so before entering into the Green Revolution, let's talk 3 options for increasing food production. Okay, now our aim is how we can increase the food production, because our population is also increasing day by day. It's booming, it's out rocketing, but the land and the resource is limited for us. So what are the probable strategies we can utilize here? Okay, so first thing is given here: agrochemical based agriculture. Now what do you mean by agrochemical based agriculture? The use of fertilizers and pesticides, right? Okay, now when you talk about fertilizers and pesticides, they are quite expensive if you want good quality of fertilizer and pesticides. And obviously overuse of fertilizers and pesticides will cause environmental pollutions. Like whenever you are you are putting fertilizers or pesticides for the growth of your crops, but indirectly they have multiple side effects. They will create a loss of biological components in soil. They will kill the microbes or the beneficial or the symbiotic organisms. Some pesticides, they will kill the earthworms. They are called for the friend of farmers. Even overuse of fertilizer will cause dehydration of your soil particles, and ultimately within 3 to 5 years your soil will become infertile. It will lose the fertility, right? And eventually during the time of raining, if these fertilizers or pesticides, they will bleach or seep into the nearby water bodies, they will harm the aquatic flora and fauna. Okay, so that is the environmental impact of overuse of fertilizers and pesticides. Okay, now let's talk about the next probability. So if you are done with agriculture based upon fertilizer and pesticide, if you don't want the utilization, then the next thing that is for you, it is organic farming, organic agriculture. But again, for countryside farmers, this organic farming is too much expensive. They cannot afford organic farming. So basically, whenever in mall or in shopping marts, whenever you will go to buy vegetables, then you will be finding there are two racks, two different types of vegetables are there. Normal vegetables, which are comparatively cheaper, and the second part is about organically farmed vegetables. They are quite costlier. Okay, so we have to opt for a third strategy, that is the use of genetically engineered crop or genetically engineered crop based agriculture. Here we will be talking about GMO, genetically modified organisms. Okay, so when we are talking about GMO, it uses genetically modified crops or genetically modified organisms are the plants, bacteria, fungi, animals whose genes are altered by manipulation. And you know that how to manipulate or alter the genetic makeup, and obviously when you are altering the genotype of an organism, that will produce alteration in phenotypic expression, right? So when we are talking about eco-friendliness, when you're talking about eco-friendly agriculture, when we are talking about sustainable, then you have to go with GMO rather going for organic farming, rather going for agriculture based upon chemicals like pesticides and fertilizers. Okay. Now let's talk about the difference between green revolution versus genetically modified organisms. Okay. Now basically green revolution, it's a kind of old concept, and genetically modified, that is a part of modern day biotechnology. So green revolution succeeded in tripling the food supply. Here we will be talking about the quantity. But when you talk about quality, we have to go with genetically modified organisms. Better management practices and agrochemicals are not affordable in developing countries. Okay. Now, if you ask, sir, which are developing countries, the country like India, Sri Lanka, Bangladesh, Nepal. So these kind of countries are called developing countries. Japan, USA, UK, France, those countries are considered as developed countries. So farmer of those countries can afford the high cost of agrochemicals if they are going with green revolution. But the countryside farmers of country like India cannot afford such costing. Okay, and you already know that conventional breeding techniques are not efficient enough. That we call hybridization. If you are going with hybridization, that means the conventional breeding technique to raise a superior variety, right? You know that one of the example we'll talk about, that is the indigenous cow, that is Sahiwal, is a variety of native or indigenous cow. We'll crossbreed this Sahiwal cow with the bull of Jersey, right? And the crossbreed is called as Jersey Sahiwal. That's a crossbreed or hybrid. Now, what is the advantage of Jersey cow? A Jersey cow can produce 35 liters of milk in a day. It is a huge lactation, right? But when you talk about Sahiwal cow, it can produce maximum 10 liters or 7 to 10 liters of milk per day. Quite low quality of lactation. But what is the advantage of Sahiwal cow? It can survive in this native environment inside the India, inside the farmhouses of India. But if you bring a Jersey cow from America or from USA, then it will unable to cope up with this environment. It's a tropical country, right? And eventually it will die. So that's why farmer, what they did hybridization between Sahiwal cow and Jersey bull. They got a new breed or hybrid, that is Jersey Sahiwal. Now it has the genes from Jersey and it has some genes from Sahiwal. Now it can produce like higher quantity of milk, and it can survive in this tropical environment of India. But obviously, due to the effect of bad genes, when you are when you are following the conventional hybridization techniques with good genes, the bad genes will also be inherited, and that will cause deterioration in the milk production in Jersey Sahiwal cow. It will produce now 15 to 20 liters of milk per day. It will not produce milk like the father or the breed Jersey, take 35 liters of milk per day. That is due to the influence of bad genes. So that is the drawback, that is the negativity of traditional hybridization techniques. But if you are following genetic recombinant DNA technology or genetic engineering, you have a choice, that is gene of interest. You will cut that gene from the donor organism, or the desired gene will be taken out from the donor organism and will be incorporated into another recipient or host organism, and that host organism will produce or will express only the desired gene. And that is the superiority of genetic engineering or biotechnology. So what is the difference? If you are following traditional hybridization technique, along with good genes, few bad genes will also be inherited into the next progeny. But if you are going with genetic engineering, you are choosing only the desired or good genes inside your genetically modified organisms. Okay. And few more impacts of conventional techniques: pollution from agrochemicals. Already we have spoken about pesticides and fertilizer, how they affect the fertility of soil, how they affect the ecosystem, right? So if you are going for sustainable agriculture, If you are going for sustainable agriculture, then our option is genetically modified crops or genetically modified organisms. These are the possible solutions. Okay. Now, before knowing how to produce genetically modified crop, we'll learn how biotechnology help us in the process of micropropagation. Also, it's called as tissue culture. Micropropagation or tissue culture. Okay. Now, obviously, for that we need help of biotechnology. You need to collect any plant tissue. Now, what kind of plant tissue you will be selecting? Young buds, it may be apical buds or axillary buds, because the budding tissue, the new tissue, has the meristem. So, we will take out newly growing budding leaves from a plant, and inside the laboratory we'll cut this piece of leaf into smaller pieces. These are called explants. Now, these explants or the tissue which is taken from this newly growing bud or a leaf, they are free of any kind of contamination, or they are any virus free. Okay. Now, we'll grow these explants. So, this basically explants are nothing but they are piece of plant tissue. Now, we'll put this explant into test tubes. Right? We'll fill the test tubes with media, plant specific or tissue specific media will be there. We'll put these explants into this media with the help of proper instrument, and the environment should be sterilized inside the laboratory whenever we are working or on tissue culture, plant tissue culture. And the next thing, we have to put this test tube with this explant for incubation, so that you can provide optimal environment, optimum moisture, optimum temperature, optimum pH for the growth of these cells inside those explant, for the growth of tissue. That's why we are calling it tissue culture. And the next step after incubation, when we are observing the tissues inside are growing, they will produce some undifferentiated mass that we call callus. That is the sign of initial growth of this tissue. Then we will transfer it into petri dishes for further growth with proper culture media. There is no soil you are utilizing, so if there is no soil, there will be no chance of contamination. Soil is reservoir of multiple pathogens, so that's why you can successfully, you will be producing contamination free new plants or plantlets. And these plantlets are produced when produced inside the lab. These small plants or plantlets are also called as soma clones. These plantlets are also called as soma clones because they are identical in nature. Their mother tissue was, or these explants were taken from this leaf. That's why you are calling them soma clones. We have done culture of somatic cells. There is no utilization of germ cells. There is no concept of ovum. There is no concept of sperms. There is no concept of flower or pollen grains. So that's why these identical plant copies are called soma clones, and when they're growing, we'll call them plantlets, and then we'll put them into agricultural land. So this is the whole process of micropropagation technique or tissue culture technique. So the terminologies you need to remember here, that is explants, somaclones, micropropagation, so what do you mean by explants? The tissue part which you have taken from budding leaves or buds of apical region or axillary region, or axillary buds or apical buds. Basically, these are containing meristematic tissue. So this is the utilization of tissue culture technique, and obviously you need help of biotechnology, and the environment should be sterile, contamination free. And by utilizing this technique, suppose you got a plant which is diseased, but interestingly, the newly growing apical buds or axillary buds, this meristematic tissue, these are viral free or contamination free. These are called meristematic tissue. These are newly growing buds, and obviously this meristematic tissue, these are contamination free or pathogen free. Now if you are following this tissue culture technique from a diseased plant, you can produce healthy plants. Into a sterilized ambience, into sterile ambience, by utilizing this tissue culture. So that is one of the major application, right? Converting a diseased plant into healthy plantlets. And you are increasing the number, that's why we are calling the process as micropropagation, going against the conventional propagation or vegetative propagation techniques, right? So you got the definition of micropropagation. You have to know what is meristem, you have to know what is tissue culture, you have to know what is explants. You have to know what is soma clones. These newly growing, newly produced clones or the identical plants from this mother plant, these are called soma clones or plantlets. Okay. Now another technique we will learn here against conventional hybridization technique is called somatic hybridization. How to produce somatic hybrids? For that you need plant cells. Okay. So these two are different plant cells. Suppose this is a cell from potato and this is another cell from tomato. So from this potato you have taken this cell, and from this tomato you have taken this cell. Now you have to digest the cell wall. Cell wall digestion is needed, and for digesting the cell wall you will be utilizing enzymes called cellulase or pectinase, and these naked cells without cell wall, these naked cells without cell wall are called protoplasts. So plant cells or fungal cells without cell wall, we call them protoplast. This protoplast of tomato cell, this is protoplast of potato cell. Now we have to perform a technique called protoplast fusion. Now there are multiple fusion techniques you can use. You can utilize chemofusion. With the help of chemicals, you will fuse these two protoplasts. For chemofusion, polyethylene glycols can be used. Page, polyethylene glycols, calcium ions can be used. Okay. There are techniques called electrofusion. There are techniques called spontaneous fusion. Okay. the success rate of electrofusion or chemofusion is quite higher. So by utilizing the process of protoplast fusion techniques, we can produce hybrids. We can produce hybrid cells, and these are called somatic hybrid. And the resultant plant which will be grown from this particular somatic hybrid, that will be called pomato. A somatic hybrid plant which has some criteria of potato and some criteria of tomato. A plant which will produce tomato as well as potato. So that's how you can produce some amazing hybrid plants by utilizing few techniques from biotechnology. That is one of the application of biotech. Isn't it amazing? Okay, let's move forward. Okay, now we have to learn what are the advantages of utilizing genetically modified plants over traditionally hybridized ones. Okay, so let's start with the topic. So it makes crops more tolerant to abiotic stresses, that is cold, drought, salt, heat. One of the example I am giving, in our time when like I was 6 to 7 years old, we used to find some vegetables only in some particular seasons like broccoli, cauliflower, cabbages, tomatoes, green peas. Only we used to get into the winter season. In summer, there is no way you can ask to the vegetable shopkeeper, uncle, please give me 1 kilo of cauliflower, 1 kilo of cabbage, right? But nowadays, 12 months in every season, you can see that newly farm grown cauliflowers, cabbages, broccolis, green peas are available. These are boon of biotechnology, right? Okay. You might have heard about that oranges or capsicum, they can be grown into some temperate regions, into hilly regions, because they need special temperature, a particular temperature, particular salt, particular minerals into the soil. But when you talk about genetically modified capsicum, genetically modified orange plants, even they can be grown in tropical soil. There are few varieties of wheats, few varieties of rice are also produced, genetically modified rice, genetically modified wheats, genetically modified tomatoes, many other genetically modified crops which have crossed the geographical barrier, environmental barrier because of the biotechnology. Okay. Next, pest resistant crop reduce the use of chemical pesticides. Obviously, there's a huge costing that's a burden for many farmers, chemical pesticides as well as pollutants. So you can get rid of that cost as well as environmental pollution if you are utilizing genetically modified crops. For example, BT cotton, BT for Bacillus thuringiensis. So it's a genetically modified cotton. Gene from Bacillus thuringiensis is punched or recombined inside cotton plant. Now, if you want to grow this cotton balls won't be harmed by cotton ball worms. It will produce a pesticide kind of chemical which is called Bt toxin, natural pesticide Bt toxin. When the larvae of cotton ball worms or those moths will eat the tissue from this particular plant, by the effect of this Bt toxin, automatically they will die. So you don't have to waste money behind the pesticides, artificial ones, right? Okay, it reduces post harvest losses. Okay, a big headache was there, like in warehouse or cold store, you can potatoes can be stored, right? Even cauliflowers can be stored, but tomatoes cannot be stored, because when you talk about tomato, it will produce a special kind of enzyme that is called pectinase. The tomato will produce a special kind of enzyme called pectinase, and gradually they will degrade the cell wall of tomato, and that will rot eventually, even in cold storage. Now, there is a genetically modified version of tomato that is called flavor saver tomato. Flavor saver tomato, a gene which will inhibit the action of pectinase enzyme, and that's how in cold storage successfully you can store these tomatoes for long term. So that is the boon of biotechnology. Next, it increases efficiency of mineral usage by plants. It prevents early exhaustion of soil fertility. It will genetically modified plants are more efficient in absorbing minerals and organic materials from soil, right? Next, it enhances nutritional value of food. For example, golden rice. So in few tropical countries, there is you can observe famine kind of situation. So scientists thought they are mainly consuming rice. Rice is their staple food. So if the nation is utilizing rice as their staple food, why not increase the quality of rice? Okay, so they produced golden rice. They introduced genes from daffodil, a flowering plant, and a bacteria called Arwinia. They introduced genes from daffodil and a bacteria called Arwinia. Beta carotene producing gene. Now obviously, whenever you will be introducing this gene, the golden rice, it will get added benefit of vitamin A and iron. So this vitamin A and iron enriched rice are called golden rice. From where you got the genes? From daffodil plants and Arwinia bacteria. So isn't it amazing again? Suppose if you are eating the rice every day we are eating, that is only rich in starch, right? Okay, but if you will eat golden rice along with carb or starch, you will get vitamin A, you will get iron. It's really beneficial. Okay, now the next thing we'll talk about, to create tailor-made plants. These tailor-made plants means by utilizing rDNA technology. The plants which are produced by using recombinant DNA technology to supply alternate resources like starches, fuels, pharmaceuticals to the industries. So whole lot of utilization of biotechnology in pharmaceuticals, in fuel industry, in food science, in medical science, What else you need? Okay. Now we'll try to understand the science behind production of pest resistant plants. Okay. So what are the benefits of pest resistant plants? They act as biopesticides, right? Cost effective. It reduces the need for insecticides, again cost saving, cost cutting or cost saving. It will replace biopesticide, act as natural biopesticide from the organism itself, and obviously you don't have to buy extra pesticides or insecticides, and obviously if you are following them, you are performing eco-friendly agriculture, sustainable agriculture. Some examples are given here, that is BT cotton, first GM crop introduced in India, first genetically modified crop introduced in India. Few other, BT corn, BT rice, BT tomato, genetically modified potato, genetically modified soybean. Even few genetically modified crops are utilized as edible vaccine. Few genetically modified potato, not BT, few genetically modified potato, few varieties of genetically modified tomato, we utilize them as edible vaccine. Few GM crops are also utilized as edible vaccine. Okay, now we'll focus on how to produce this BT cotton. What is the importance of this BT, from where we got these letters? Let's talk about that. Okay, so I'm summarizing it. So there is a bacteria called Bacillus thuringiensis, that produces a toxin called BT toxin. Basically, these toxins are kind of delta endotoxins produced by some crystal proteins or cryoproteins. That is a kind of immunological phenomena observed in this Bacillus thuringiensis bacteria. They protect themselves from insect attack. So in nature there is a tendency of some specific larva of insects, they will feed upon this bacteria, Bacillus thuringiensis. But in adverse condition, if you feed upon the population, they will die. So that's why they have developed a toxin that is not harmful for bacillus, this BT toxin, that will not create harm to this bacillus, but obviously that will exert damage or harm to the digestive tract of this larvae of insects. They will not kill adult one, they will kill the larvae of insect, because the tendency of the larvae, they are very much voracious. The larvae of insects, they tends to eat the tissue from inside the birds, inside the leaves, inside the meristem, right? The vascular tissue, they will like destroy the plant, and that will cost the farmer. So that's why if you are utilizing genetically modified crops, and if you are tagging the gene, BT toxin producing gene from Bacillus thuringiensis into your crop, it will automatically act as biopesticide. Okay, so let's talk about, so the topic is BT cotton. BT cotton, the first introduced genetically modified crop in India. Some strains of Bacillus thuringiensis or Bacillus thuringiensis. Some strains of Bacillus thuringiensis have proteins that kill insect like coleopterans, Lepidopterans and Dipterans. Okay. Now, why it's so? If this is the bacteria, Bacillus thuringiensis, shortcut, we are naming it BT. The shortcut name of Bacillus thuringiensis is BT, right? Now, there is a tendency of these larvae of certain insects, LCD, Lepidopterans, Coleopterans and Dipterans. LCD is a tree. Okay. Now, larvae or larva of these three insects, LCD, Lepidoptera, Coleoptera and Diptera, they try to feed upon this bacteria, BT, Bacillus thuringiensis. Okay. Now, if you observe their growth stages for bacteria, there is a lag phase, there is exponential growth phase or log phase, there is a stationary phase and there is a decline or death phase. So, there are growth phases called lag phase, log or exponential phase, stationary phase and death phase. Okay. So, basically, when they reach stationary phase, when they reach, in NCERT is written a special phase of their growth, when they reach into stationary phase, and if these larvae are attacking or feeding on Bacillus thuringiensis, in order to survive, they will produce special protein coded by Cry gene, crystal protein. They will produce Cry proteins. These are inactivated toxins. These Cry proteins are called protoxins. They are not activated toxins. That's why they cannot harm the bacterial colony. Whenever this larvae or larva will eat that, due to the alkaline, if they come into contact with alkali or alkaline environment, they will get dissolved with their digestive juice and this protoxin will get activated and will form toxin, and these toxins are called BT toxin, and this BT toxin that has ability to kill this larva. BT toxin has ability to kill the larva of of those particular group of insects. Remember LCD, Lepidopteran, Coleopterans and Dipterans. Again, which all groups of insects? Lepidopterans, L, C, Coleopterans, D, Dipterans. You know, there is a particular kind of TV called LCD TV, particular kind of lights called LCD lights. Okay. Now, which insects are under Lepidopterans? You need to study these as well. Question can be come from anywhere. Question can be curated from anywhere. Lepidopterans, examples are tobacco budworms, and armyworms. Now, here is a trick: laptop army. Laptop army. This is the trick. Lap for Lepidopterans, top for tobacco budworms, and who is utilizing this laptop? Army, that is army worms. Okay. Now see, coleopterans. Coleopterans, examples, beetles. Coleopterans, example, beetles. Okay. Now, what is the mnemonic? Cold beer. Cold beer. So you can imagine like this: army sitting with laptop, Lepidopteran, tobacco birdworms, and army ones sipping cold beer. There is coleopterans, and examples are beetles. Now LCD, last one, dipterans. Who are dipterans? Flies and mosquitoes. Flies and mosquitoes. So di means two. There are two examples, flies and mosquitoes. These are under dipterans. Okay. Now let's resume our learning. So this Bacillus thuringiensis forms an insecticidal protein that is called Bt toxin crystal during a phase of their growth. So in which phase of their growth? Stationary phase of their growth. Stationary phase of their growth. It does not kill the bacillus as it exists as inactive protoxin. In order to activate it, you need to dissolve it into alkaline solution. Okay. Now, how that will become activated? When an insect ingests the toxin, basically the larva of that insect, it becomes active due to the alkaline pH of the gut. So the pH of insect is always more than 9. It's quite alkaline, right? So even if you consume this toxin, that won't be harming you, because the highest pH of your gut, of your alimentary canal, cannot be 9. It will be always lower than 9. It will maximum 7.8, maximum 8.5 or 8.6. But the gut pH, particularly the mid gut pH of insect, always higher than 9, quite highly alkaline or basic in nature, which solubilize the crystals, and now this toxin binds to the surface of midgut epithelial cells, where they will be digesting and absorbing the digested food. And now when this toxin is binding with the epithelial cells, that will create pores, that will digest, that will destroy the cells and create pores into their midgut, and it cause cell swelling and lysis and death of that insect, because they are unable to absorb the digested food, because the midgut cells are damaged and degraded. Okay. Now, Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into crop plants such as cotton by utilizing rDNA technology, recombinant DNA technology. Most Bt toxins are insect group specific. They are coded by which genes? Cry genes, crystal forming genes. Okay. Now, how you will perform or you will tag the cry gene into your targeted plant or into your host organ? By utilizing vector, that is Ti plasmid, and for that you have to use a bacteria which can infect plant cell, that is Agrobacterium tumefaciens, the natural genetic engineer that always act as the vector for plant cells, Agrobacterium tumefaciens. Okay. So proteins encoded by Cry1AC and Cry2AB genes control cotton bollworm, and protein called Cry1AB gene controls corn borer. Now, how to remember this? When we are talking about cotton bollworm, then the gene will be coding the cry protein, cry1 AC and cry2 AB. Cry1 AC and cry2 AB genes control the cry protein production into and we use these cry proteins produced by these particular two genes to control cotton bollworms. Now, how to control corn borer? Cry1 AB. Okay. Now, there are many tricks are available. One of the trick I am telling. So, basically cotton bollworm, the length of this name is more than corn borer, and cry 1 AC. So, cry is common in between them, so omit that. So, if you are talking about 1 AC, so that is 1, A is the first alphabet, C is the third alphabet. So, summation will be 3 plus 1 plus 1, that is 5. Cry to AB, 2 plus 1, A is the first alphabet, B is the second alphabet. 2 plus 1 plus 2 equals to 5. So, when the summation is coming as 5, you can consider this is for the larger one, that is cotton bollworm, and for corn borer kind is shorter than cotton bollworm. So, let us decode this cry 1 AB, 1 for 1, A for first alphabet 1, B for second alphabet, that is 2, and summation will be 2 plus 1 plus 1, that is 4. So, 4 for cry 1 AB, corn borer, cry 1 AC 5, cry 2 AB 5 is for cotton bollworm. So, we are done with this trick. Now, let us move forward. Okay. So, few diagrams of cotton boll. You can also call it cotton ball, BALL, but more precisely it is said as cotton boll, BOLL. So, these are cotton bolls. These are affected by bollworms. Basically, this is the moth. This is a kind of moth. The larva is the attacking stage. Here you can observe the larva of this cotton bollworm attacking this cotton boll and destroying the tissue inside, and obviously that will harm into the production of cotton. And if you have a GM crop, that is BT cotton, when the larva will start eating the ball, it will produce the cry. The larva, when it's trying to digest the cry protein, it will damage the midgut of the larva, and they will die automatically. Okay. Now, next example we will talk about. Okay. So, classic example of application of biotechnology, that is nematode resistance in tobacco plants. So, let me tell you the story. So, there is a nematode or ascalminth called Meloidigyne incognita. Okay. Now, this nematode, the larvae, they are much voracious. they will try to attack the root. They will enter into this tobacco plant through the root, and they will try to eat the vascular tissue inside the root, right? Now when the vascular tissue is damaged and they are establishing their colony inside the roots of tobacco plant, that will create tuber kind of structure into the root, or nodule kind of structure into the root. We call them root knot, and once the root is damaged, the plant will die due to the root knot disease, right? Okay. Now in order to stop the attack of this particular nematode, we'll take the help of recombinant DNA technology. We'll utilize genetically modified tobacco plant by utilizing a special technique called RNA silencing technique or RNAi technique, right? So let's talk about that RNAi technique or RNA interference or RNA silencing strategy. So a nematode called Meloidigyne infects the roots of tobacco plants, causing reduction in yield by causing that disease called root knot. Okay. Now it can be prevented by a special technique called RNA interference strategy or RNAi or RNA silencing. So our motto is to silence the process of translation. So what is the central dogma inside the cell? DNA to mRNA, mRNA to protein. Now if you silence the mRNA, there will be no translation, there will be no production of protein, and if there is no protein, ultimately if our organism fails to produce a certain protein, that may die or reduce the activity. So that is our motto. So RNAi is a method of cellular defense in all eukaryotic organism. When you talk about prokaryotic organism, what are the cellular defense mechanism? Yes, for prokaryotic cells like for, E. coli, it uses methyl transferase enzyme, methylation of DNA technique, and restriction endonucleases, particular kind of enzyme, right, to cut the infected or the foreign DNA inserted into the bacterial cells in order to stop the viral attack. So that is the cellular defense mechanism for prokaryotic cells. What are the cellular defense mechanism? They use particular enzymes, restriction endonuclease and methyl transferase to block the attack of viruses, particularly the phage virus, bacteriophages. Now for eukaryotic cell, the cellular defense mechanism is called RNA interference technique. So what it does? It prevents translation of a specific mRNA, that is called silencing, due to a complementary double-stranded RNA. This is the most important thing you need to remember. By utilizing double-stranded RNA molecule, we are performing mRNA silencing by utilizing a double-stranded RNA. Now, what is the utilization or importance of double-stranded RNA? Obviously, we are going to discuss. Okay, so let's talk about that. So here a few images are given here, few pictures are given here. You can observe here that healthy tobacco plant, right, healthy roots, but when it's affected by Meloidigyne incognita, a special kind of nematode or ascal, means how it's affecting the plant, right? So this is the roots of diseased plants. We call these as root knot disease. And this is our Meloidigyne incognita. They are forming burrows or trying to penetrate inside the roots of tobacco plants. So this is the Meloidogyne incognita, paste to plant or parasite, endoparasite of plant. Okay, so these are the cells of the root of tobacco plant. You can observe here how it's penetrating inside and causing the root knot disease. Okay, so we'll talk about the importance of the double-stranded RNA. So if this is a cell, suppose this is a cell, eukaryotic cell, cell of any plant. Now when a virus will, how a virus attacks eukaryotic cell, a virus inject, suppose the virus inject double-stranded RNA, dsRNA means double-stranded RNA inside the cell. Now this virus will also inject reverse transcriptase enzyme to convert this double-stranded RNA into double-stranded DNA. By the process of reverse transcription, and this DNA will produce mRNA by the process of transcription. Now mRNA will take the help of ribosomes, and by the process of translation it will produce proteins. Now as because the virus has hijacked over the genetic makeup, a central dogma of this eukaryotic cell, the proteins will be of the virus, that is viral proteins or viral parts. Now these viral parts will get assembled inside the cell. This virus, what it will do? It will rupture the cell and will attack the neighboring cells. So this is how a virus is affecting cells and also spreading the infection. Okay. Now our motto is to block this mRNA. If you get some technique to block this mRNA, block this translation, then no viral parts or no viral proteins will be produced here. So that's how the RNA silencing or RNA interference technique works. Okay. So what you have to do, you have to incorporate, you have to input double-stranded RNA inside the cell. Now I am talking about this RNA silencing mechanism. Yes. Now this double-stranded RNA, what it will do? It will get recognized by a special complex of enzymes called dicer. Dicer complex will recognize this double-stranded RNA molecule and break it down into smaller pieces. We will call them siRNA, small interfering RNA. Now this siRNA will get recognized by another complex of enzymes called RISC, risk factor, and risk factor will sit on this small interfering RNA or siRNA. And remember, this double stranded RNA has two strands. One strand is called as sense strand and another strand is called as antisense strand. Now remember, this is the sense strand which can code for protein. Antisense strand is complementary to the sense strand, but when it will bind with the sense strand, it will inhibit the activity. Okay. Now our double stranded RNA got a sense strand and another antisense strand. Now this risk factor will sit upon the sense strand and it will cleave sense strand into pieces. It will cut sense strand into pieces. So ultimately what we got, a single stranded RNA strand, that is the antisense strand. So this is the mechanism of RNA interference. Now how this antisense strand will interfere? If you get this antisense strand, now while this viral double stranded RNA has produced this mRNA that is actually a sense strand, and what you have to do, you have to tag this antisense strand. The cell will tag this antisense strand with this mRNA and now the translation will get blocked. Now this translation will get blocked. During the time of post-transcriptional modification, this mRNA will get attached with this antisense strand which is complementary to each other, and that's how translation will be blocked and there will be no formation of viral proteins or viral parts. Now you will ask me, sir, how this double stranded RNA which is responsible for RNA interference, how cell or that particular eukaryotic cell, how it got that double stranded RNA which is acting as a savior for it. Now during the normal infection or during the influence of some mutation or transposition or jumping genes, few parts of cell will get modified, few part of central dogma will get modified and will form this kind of severe double stranded RNA, ultimately which will produce antisense strand upon action of Dicer complex and RISC complex, and this antisense strand will block the mRNA and translation which cell got during the time of infection from the virus. I think now you got the whole concept. Okay. So let's see what NCERT is saying. So the source of this complementary RNA, now the question can be asked, from where cell got this double stranded RNA which is responsible for RNAi? So source of this complementary RNA is from an infection by any other RNA virus or some mobile genes or jumping genetic elements called as transposon. Remember, these transposons are also called as jumping genetic elements. During the time of any infection, generally we get this kind of genetic material. Okay, so that replicate via an RNA intermediate. Now for our genetically modified tobacco plant, what we have to do now? We have to isolate that nematode specific genes, that is DNA. It is introduced into the host plant, that is tobacco plant, by utilizing a vector, that is agrobacterium, and the plasmid will be TI plasmid. Okay. Now it produces both sense and antisense RNA in host cell. So we have to choose two different genes, one piece of DNA that will produce the sense strand, another piece of DNA that will produce the anti-sense strand, that is called anti-sense technology. So that in host cell, when we will be putting that recombinant DNA, it will produce both the strand of mRNA, one sense and another anti-sense strand, and as because they are complementary to each other, they will get bind to each other to form double-stranded RNAi RNA. So these RNAs are complementary, so they form double strand RNA. It initiates the RNAi and silences the specific mRNA of nematode. Thus, the parasite cannot survive in a transgenic host expressing specific interfering RNA. Okay, now let's see how it's affecting the parasite. Now, if you understood this concept, now let's jump to this one. Okay. Now, what we have done? We have inserted specific DNA sequence inside the root cells of the tobacco plants, right? Now, inside the root cells, those DNA producing sense strand of mRNA and another DNA segment which will be producing antisense strand of RNA that will produce the double-stranded RNA inside the root cells. Now, while this myeloidogyne will feed upon these cells, when the myeloidogyne, the nematode feeds upon the cells of the vascular tissue of that tobacco plant, it will produce a voracious protein inside that is responsible for the growth, or a growth factor is produced inside the myeloidogyne. So our motto is to inhibit that growth factor so that myeloidogyne eventually will die. Okay. Now after feeding plant roots, it will get the double-stranded RNA from the plant cell. Now this double-stranded RNA will get incorporated into the cells of this myeloidogyne larva, this myeloidogyne incognita larva. Now once this double-stranded RNA from the host plant is coming inside the cells of myeloidogyne, the RNA silencing process will be initiated. Inside that myeloidogyne, the dicer processing will be there that will cut that double-stranded RNA into siRNA, small pieces of double-stranded RNAs. Now activation of RISC complex will be there. RISC complex will bind with the sense strand of that double-stranded RNA. It will cleave or chop the sense strand into pieces, and inside this myeloidogyne, what you are getting here, only the remaining antisense RNA are present. Now this antisense RNA will bind, this antisense RNA will bind with the particular complementary gene which is responsible for the growth of this particular, when this antisense RNA will bind with the growth factor or growth hormone producing mRNA, ultimately that mRNA is silenced. Now from normal growth there will be stunted growth. Again, if you have not understood it, I am emphasizing or elaborating it. Okay. Suppose this is myeloidogyne incognita. Whenever it's feeding upon the plant cell, how it is utilizing this plant cell for the growth. So inside the body of this larva, there is a special gene or DNA responsible for producing of one mRNA. and the product of this mRNA will be such proteins, and these proteins will help this particular nematode to produce growth factors, and that will stimulate the growth of the larva. Now our motto is to block this mRNA. Now how to block this mRNA? What we have done already, we have produced a genetically modified tobacco plant, and when this larva, notorious larva, is feeding on genetically modified tobacco, it's getting the double-stranded RNA inside its cell. Now when the cells of this myeloid gyne observing this double-stranded RNA, what they will do, from this double-stranded RNA, they will activate dicer and risk protein, and ultimately from this double-stranded RNA, the sense strand and antisense strands are there, right? They will cleave the sense strand of this double-stranded RNA, and only the antisense strand is remaining. Only remaining the antisense strand. Now this antisense strand will come here and will bind with this mRNA and will block the process of protein synthesis and translation. And if there is no growth factor, then the growth of this this nematode larva will become stunted. So this is a larger one, this has become a smaller one, stunted growth. Now if the growth is stunted generation by generation, gradually they will become eradicated or they will die eventually. So that is the whole concept. Okay. Now we are jumping to the next topic, that is application of biotechnology in medicine. Okay. So products from non-human sources cause unwanted immunological responses, but recombinant therapeutics does not have such problems. Like when you're talking about medicines, we may take help of some other animals or other organism for those therapeutics, right? Suppose you need a particular enzyme or particular hormone to control a special situation inside your body. Now for that you have to sacrifice cattles, horse, we are utilizing horse, pigs, cows, buffaloes, rabbits. We are utilizing them as organisms, we are sacrificing them, we are extracting particular enzymes, proteins or hormones from their body and applying on applying on human being. But as because we have special immunity that is very much specific towards our species, that is very much specific towards our own selves, they will try to block or create immune response or allergic response against those foreign proteins, like foreign insulin, foreign enzymes, right? So in order to avoid the side effect of those foreign proteins, we will be utilizing biotechnology to produce our own protein. We will be producing human proteins, and then if we inject those human proteins inside our body is gonna like welcome them happily. Our body's immune system will not gonna produce any immune response against them, right? So at present about 30 recombinant therapeutics have been approved. Of these, 12 are being marketed in India. You need to remember this information. 30 30 recombinant therapeutics are available or approved, and how many are marketed in India? 12 are being marketed in India. Again, 30 therapeutics are approved, 12 are marketed in India. Now we'll talk about genetically engineered insulin as our example. Okay. So insulin is used to manage adult onset diabetes, that is also called as type 2 diabetes or non-insulin dependent diabetes mellitus. Type 1 diabetes, that is IDDM, insulin dependent diabetes mellitus. Type 2 diabetes is non-insulin dependent diabetes mellitus. Now what is the difference between type 1 and type 2 diabetes? Okay, for type 1 diabetes, who can get affected? Anyone from any age. There is no age barrier for type 1 diabetes. So basically what happens, your body's autoimmunity will kill beta cells of islets of Langerhans from pancreas, and if there is no beta cells or less beta cells, so less amount of insulin will be produced. So that is called type 1 diabetes. But adult onset diabetes, that is type 2 diabetes, so here what happens, your body sometime will produce enough amount of insulin, but due to the insulin tolerance, the receptors will not respond against that insulin produced inside our body. So in that case, artificially we have to inject insulin to recover that insulin tolerance. Okay. So insulin from pancreas of animals like cattle and pigs causes allergy. So in old or conventional practice, it's not possible to sacrifice human being and to extract the pancreas and to extract the beta cells from pancreas to produce insulin, human insulin, right? So what they used to do, they used to slaughter cattles and take out their pancreas, and that cattle insulin is almost similar to human being, though it will work, but that will cause a side effect like allergic response, immune response will be provoked. So in order to stop that, we have to take help of biotechnology. Okay, so now it is possible to produce human insulin, we call it humalin, by utilizing E. coli bacteria. Okay, insulin consists of two short polypeptide chains. There is chain A and chain B. One is of 21 amino acid and one is of 30 amino acid chain that are linked by disulfide bridges. Okay. So in mammals, insulin is synthesized as prohormone or proinsulin, that is kind of inactivated insulin. Now it need processing, post transcriptional modification, protein chain, right? So it needs post transcriptional modification or maturation to become matured and functional insulin hormone. Now the prohormone contains an extra stretch of polypeptide chain called C-peptide. So this is removed during the maturation into the insulin. So proinsulin has three chains, A chain, B chain and C chain, connected through disulfide bonds. Now upon maturation, or post translational modification, the insulin we get that has only A chain and B chain, C chain is cleaved. So this is what I was talking about. This is the proinsulin. This is the proinsulin. This is the A peptide chain. This is the B peptide chain. And this particular stretch, which is joining A to B, that is called C peptide chain. Now this is during the time of maturation. Pro-insulin convertase, post-translational modification is happening here, and we got this active insulin which has A peptide and B peptide. C peptide is cleaved out, or free C peptide. So this is the normal mechanism of biosynthesis, biosynthetic mechanism inside our cells, right? Now we have to perform rDNA technology in order to produce human insulin. Okay, we have to produce genetically modified organism to produce human insulin. So what you thought? You have taken the insulin producing gene. You have taken insulin producing gene from human being and directly incorporated into the genome of E. coli bacteria. So that E. coli bacteria will produce pro-insulin, because the gene which is coding inside human body, that is producing mRNA, and that mRNA is producing pro-insulin, not the insulin. But in artificial condition, it is not possible to perform the post-translational modification, or it's very much complex technique for us. So there is a problem. When they wanted to perform rDNA technology or genetic engineering to produce human insulin or humalin, this was the problem they faced. Because if you directly taking out the gene, that will produce pro-insulin, not the insulin. And in artificial condition, in laboratory, it is not possible for us, or it's very much complex to produce insulin from pro-insulin. So there is a solution for that, and that was given by the company, medicine company called Eli Lilly. So what they did in 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A chain and B chains of human insulin, and then introduced them into the plasmids of E. coli to produce insulin chains. Now chains of A and B were combined by creating a disulfide bond to form human insulin or humalin. Okay, so tactfully they did this work. They coded a specific DNA which will produce A peptide chain, and they have coded a separate piece of DNA which will produce B peptide chain. So separately, with the help of two different sets of E. coli bacteria, they had produced A peptide chain and B peptide chain. Then in laboratory, they will fuse them with disulfide. They will bind them, not fuse them. They will bind them with disulfide bond. Now when you are binding A chain and B chain of insulin with disulfide bond, that will produce a perfect insulin molecule which are produced inside our cells upon post-translational modification. Okay, so that's why they had taken two different plasmids of E. coli bacteria. Okay, they had inserted insulin gene for producing A subunit inside the LAGZ gene, and insulin gene for B subunit inside the same LAGZ gene or beta galactosidase gene. Upon this insertional inactivation, they produce two different strains of genetically modified E. coli. Now this E. coli will produce A chain. This different E. coli will produce the B chain, but these chains are fused with beta galactosidase protein. So what they have to do, they have to purify A chain and B chain from beta galactoside protein chain. Now they will chemically remove A chain and B chain and combine them by preparing or bridging them with disulfide bond and got human insulin or humalin. Okay. Now next thing is about gene therapy. Now what do you mean by gene therapy? We know that we got some congenital disorders. There are few diseases which are called acquired disease, few diseases which are for our inheritance pattern. We got defaulty genes from our parents. Now in that case, normal therapeutics will not help us. We have to go for we need to seek help for biotechnology, and we have to go for gene therapy. Okay. So it's a method to correct a gene defect in a child or embryo, right? Here genes are inserted into a person's cells and tissues to treat a hereditary disease or genetic disorders. It compensates for a non-functional gene. So there was a mutation, right, in the genetic makeup of that person. So that mutation switched off the process of production of some functional proteins. Now when you have inserted that recombinant DNA inside that rDNA inside that child or inside that embryo, that defect is being corrected now. Okay. Now we'll need to talk about the first clinical gene therapy that was performed in 1990, was given to a 4-year-old girl with adenosine deaminase deficiency. Now let's talk about this particular deficiency. First gene therapy performed in 1990. This little child had a rare disease called adenosine deaminase deficiency. Okay. Now what if you got the disease, then what kind of problems you are gonna face? So adenosine deaminase, basically that's an enzyme in our body. Deoxyadenosines are produced. They are quite harmful for us. If your body keep accumulating deoxyadenosine, then our T lymphocyte will fail to mature their prime cells of our acquired immunity, and if T cells are failing to get matured, then obviously your immune system will fail and you will get a disease called severe combined immunodeficiency, SCID, severe combined immunodeficiency disease. Okay. So if you don't have this ADA, adenosine deaminase inside your body, if your body is failing to produce that enzyme, you will get severe combined immunodeficiency, SCID. Now what this ADA does, it will split this deoxyadenosine into deoxyadenosine and hypoxanthine, and gradually from this deoxyadenosine your body will produce uric acid, phosphates, water, and energy is released. From hypoxanthine your body will produce 2-deoxyribose, 1-phosphate, and this toxicity will be removed from your body, and that will give a boost to your T lymphocyte and your normal immune function will be protected. So basically thing is that you don't have to learn all this process. What I am trying to tell that it's about ADA, adenosine deaminase enzyme. So this adenosine deaminase gets accumulated into your body, that will create toxicity, and that will block T lymphocytes for maturation. And if you don't have T lymphocytes into your body, if the T lymphocytes are dying or not maturing, then your immune system will face a failure. But if your body is producing enough amount of adenosine deaminase, then these deoxyadenosines will get broken down and your body gradually removes the toxicity, right? Now, how to cure that situation? Okay. Now, this is caused due to the deletion of a gene of adenosine deaminase, an enzyme for the functioning of immune system. Okay, it can be cured. Now question is how it can be cured. There are three ways. First, by the process of bone marrow transplantation. If you are a child, then we have to opt for bone marrow. Entire bone marrow will be transformed, right? Transplanted. Why it's so? Because what is the production site of T lymphocyte? The primary lymphoid organ, that is bone marrow. Production site of T cells and B cells and all blood cells. The myeloid stem cells are produced inside our bone marrow. So you need to replace entire bone marrow. Okay. Or it can be tackled by enzyme replacement therapy, by injection of adenosine deaminase. Like if you have diabetes, you are taking injection of insulin. Similarly, that's a hormone basically, but this is an enzyme. If your body don't have this ADA, you have to take injection of this particular enzyme. This is the second methodology. And the third methodology, treating the embryo. Third methodology will be treating the embryo. Okay. Third will be bone marrow transplantation, enzyme replacement theory. Okay. Embryonic cell transformation. You have to transform the cells of embryo. So that is the most effective one, right? If you want to treat the disease from the root, you have to treat the cells of embryo, or it will be best if you can treat the embryo in when the embryo is in blastula or morula stage, the cells we call blastomeres. Okay. Then if it's not detected at that time, then obviously bone marrow transplantation can be done. Okay. Now let's observe how to transplant this bone marrow. So gene therapy for ADA deficiency. So collect lymphocytes from the patient's blood or bone marrow, grow in culture, introduce a functional ADA complementary DNA into the lymphocytes using a retroviral vector. So you will be transforming the lymphocytes by utilizing vector retrovirus. Disarmed retrovirus will be used here. Okay. Now they are returned to the patient. So this should be periodically repeated as lymphocytes are not immortal. If the ADA gene from the marrow cells is introduced into the cells at early embryonic stage, it could be a permanent cure. You need to remember that. If you want to get the root of the disease, you have to cure the embryonic cells. But for the first instance which was performed for this 4-year-old girl child, so what we did that time, taken out plus patient's blood or T cells or bone marrow cells. Okay. will transform this cell with utilizing disarmed virus vector or retrovirus. Now this virus vector, what it will do, it will inject the gene for producing ADA inside the lymphocytic cell, will culture the T cells again into the laboratory to increase their population, and then will inject the T cell into that little baby. Okay, but you have to perform this process again and again and again, because after a few days the T cells will die, right, because they are kind of WBC. So it's quite hectic and hideous job. So that's why the best option is to go for embryonic stem cell therapy. Okay, now another boon in medical science, that is molecular diagnosis. Okay, now we'll try to differentiate between some conventional techniques and biotechnology. So conventional methods like serum and urine analysis are not suitable for early diagnosis of disease. So generally when we fall ill, we visit doctor, they give us urine test, blood test, right, stool test, mucus test, serum test. So that will take time. Sometime it will take 1 day, sometime that will take few hours, sometime that will take even 5 to 6 days to get the report. Okay, so that may cause a delay into the treatment. So it is possible by techniques such as recombinant DNA technology or rDNA technology like PCR and ELISA, autoradiography, southern blotting, probing techniques. So multiple techniques are there. So if you can remember, during the time of that COVID, if you are like traveling from one place to another place, into the rail station and into the airports, they used to check your smears from of your saliva, and within 10 minutes they used to give the result by performing RT-PCR, reverse transcriptase PCR. And PCR is one of the principal techniques in biotechnology, we all know that. So instead of going for the conventional pathological studies, we can introduce our modern techniques of biotechnology for very frequent and rapid disease diagnosis, right. So let's talk about PCR, polymerase chain reaction. Okay, so very low concentration of a bacteria or virus can be detected by amplification of their nucleic acid by PCR. So basically that is the, if you have one copy, one page written, now you need to distribute that same page among 10 students. What you will do, you will go to the Xerox, you will perform the Xerox copying by utilizing a Xerox machine that will produce multiple copies, and you will distribute among students. So similarly, this is the biological genetic Xerox machine, that is thermal cycler machine of PCR polymerase chain reaction is called thermal cycler. Machine of PCR is called thermal cycler. By utilizing that machine, you can amplify the sample DNA, right. Within hour you can produce billions of copies of that same DNA. Okay, that's the importance of PCR. Now where you are gonna use this PCR? To detect HIV or suspected patients, to detect any retrovirus infection, to detect gene mutations in suspected cancer patients, to identify many other genetic disorders, right. Now let's understand how this molecular diagnosis along with PCR and ELISA works for us. Okay, so basically what we do, you got a DNA sample, or RNA sample. Now we will give it to the laboratory for PCR. Once the PCR will be performed, we will get the DNA is amplified, means you got billions of copies of this DNA. billions of copies of that same DNA you got now. That is called amplification. What your cell is doing every time by the process of DNA replication, that is artificially we are replicating the DNA. Okay. Now next what you have to do, we have to cut the DNA with the help of RANs, restriction endonucleases. When these DNA pieces are separated into smaller pieces, we have to separate them out according to the based upon their size by performing gel electrophoresis. The process of separation of DNA fragments. Now what after gel electrophoresis? We have to perform southern blotting. Basically, we will take impression of the gel bands into a nitrocellulose membrane. Then, upon the sheet of this southern blotting, that nitrocellulose membrane, you got the impression of the DNA. Now you have to perform some denaturation of DNA. These DNAs are double stranded, you have to convert them into single stranded DNA. Then you have to perform probing technique. Here you need radioactive probes. After the process of hybridization, now the single stranded DNA sequence which is complementary to our genetic makeup will be applied to this nitrocellulose membrane, that is called autoradiography. Now upon performing the autoradiography, you can go for identification. So it's not only PCR, we need help of gel electrophoresis, we need help of southern blotting technique, we need help of autoradiographic technique to conclude that what kind of mutation or disease is present, right? Okay, so these probes will start binding with these DNA strands. Now, in certain cases, the probes will not bind into particular DNA. So that is the signal. The probes are complementary to our own DNA, and if it's failing to bind with the DNA present into that gel sheet, that means there is some foreign DNA or genetic mutation is there. So how it will appear if the probe is successfully binding with the DNA strands, it will produce the radiation as because they were the radioactive probes. And if there is absence of radiation in a particular place, so that indicates gene mutation or something foreign, which the probes are unable to identify and bind with that. So that means if all the DNA strands are occupied by probes, then you are absolutely healthy. If so few DNA strands are unable to bind with the probe and unable to give the radiation, that means mutation is there, probable diagnosis of particular disease, maybe it's HIV, maybe it's COVID, maybe it's any other retroviral disease, maybe it's cancer, maybe something else. Okay. So this is how the whole process is done. Now we'll jump to another process which is also utilized for molecular diagnosis and very rapid technique, that is ELISA, enzyme linked immunosorbent assay. The process of ELISA, in higher classes we will learn few other techniques, we will learn about RIA as well, radio immunoassay. So when you talk about enzyme linked immunosorbent assay, it's purely based on antigen antibody interaction. So if you are infected by any disease, your body will start producing antibodies against that, right? Because the foreign pathogen is entering into your body, that is producing antigen. What? Let me repeat. When this foreign, suppose this is the bacteria, this is the pathogen. When this pathogen is entering into your body, entered into our body, infection is there. So inside our body, it will start, that bacteria, it will produce some toxins. These toxins are called as antigens. Now you have your immune cells inside your body to protect you against these attacks. Now if you have B cells, you know about their function, they will produce these Y-shaped proteins and we call them antibodies. In order to neutralize the attack of antigens. So ELISA is based upon, with the help of ELISA, with the help of special enzyme and chromogenic substrate, we have to identify presence of antigens or antibody inside our body. So let's see how it happens. So infection by pathogen can be detected by the presence of antigens, proteins or glycoproteins, or by detecting the antibodies synthesized against the pathogens. So important thing here, for ELISA you can opt for detection of antigens as well as you can opt for detection of antibodies. Both kind of ELISA, different types of ELISA are available. Okay, so here with our ELISA we will try to identify if there is presence of pathogenic or antibody produced at the response of pathogenic attack, right? So ELISA plates are already coated with possible antibodies, right? Now suppose I have taken your blood or taken the serum from your body. So what I will do, if you are infected by a particular disease, then obviously the antigens are present here. The antigens are present here. So we are basically detecting the pathogenic antigens into our body. So if you put blood into that ELISA tubes, then that antigens will go and bind with specific antibodies present already on the wall of these ELISA plates. Okay. Now what after that? Now you have to identify, you have to prove that yes, there is successful binding of pathogenic antigen with my antibodies in ELISA plates. Next, you have to add substrate, you have to add few other antibodies. Now, when the successful binding of antibodies with the disease causing antigen will be there, if the binding successfully is taking place, few enzyme will get activated inside the ELISA plate, and in the presence of chromogenic substrate, the color of the tubes will change. And if there is change in color inside the ELISA tubes, then is a sign of identification of specific antigen. Let me elaborate it if you have not understood this. Okay. So already in our lab, we have this kind of ELISA tubes are there. Now, if you enlarge the surface of these ELISA tubes, what you are gonna get? This surface are coated with specific antibodies for the suspecting disease. Okay. Now, next what you have to do? You have to collect blood from patient, right? Now, you will put this collected blood into this ELISA plates, and if that blood has the antigenic proteins, they will bind with the antibodies present already inside that ELISA plate. Now you need some confirmation that yes, the antigen antibodies successfully binded with each other. Now here you need some special antibodies tagged with enzyme, and also you have to put chromogenic substrate. Now what will happen now? These enzyme tagged antibodies, will bind with the another surface of the antigens, and as because you have added chromogenic substrate, if there is successful binding occurred inside the ELISA plate, the enzyme will react with the substrate and that will change the color of that ELISA plate, and that is the positive signal that that person has that particular disease. I think now you got the concept. Now let's move forward to the next topic, that is transgenic animals. Transgenic animals. Okay. So these are animals whose genome has been altered by introduction of a foreign gene by manipulation. Use of RNA technology on animals. Okay. Transgenic rats are there, transgenic rabbits, transgenic pigs, transgenic sheep, transgenic cows and fish. Okay. Over 95% of the transgenic animals are mice. The most important line from transgenic animals of this chapter. There are many transgenic animals can be produced, but we say that rat race, rat of lab or mice. So whenever we are performing laboratory activities as model organism, when we are choosing animals, most of the time we will be utilizing mice. Apart from mice, other animals like rabbits, pigs, sheep, cows and fish, Drosophila, uh, C. elegans also can be used for different study in genetic engineering, right? But the most important, over 95% of transgenic animals are mice. Because why mice? Because they have very short lifespan, easy to be raised inside the laboratories. They don't need large space, they don't need special care. Okay, easily their DNA or the genetic configuration can be altered. Even they're very much easier to use in biotechnology lab. They will be easily adapting the human genes. Okay. So what are the different benefits of utilizing transgenic animals? To study the regulation of genes and their action on normal physiology and their development, that is study of insulin like growth factor. So for your research or for your study, you really cannot use human models, right? You cannot apply something. If you are doing something, if you are coming with a new drug, new cosmetic, directly you will not get permission if you want to apply on human being. So that's why for that you have to go with trial on animals. To study the contribution of genes in the development of a disease and thereby new treatments, that is transgenic models for human disease. Transgenic models for human disease such as cancer, cystic fibrosis, rheumatoid arthritis and Alzheimer's. So if you want to study the detailed procedure of disease development, okay, and how they are influencing the physiology, really you cannot observe inside a human being. Nobody will volunteer said that please apply this on me, right? So that's why we will be utilizing some transgenic animals, will genetically modify them, will utilize the human gene and will transform their cells and will observe the disease development inside their body. Okay, biological products like human protein, one of the example, alpha-1 antitrypsin used to treat emphysema. Alpha-1 antitrypsin. Emphysema is a chronic disorder you will get from chronic cigarette smoking, right, or from pollution that will cause reduced alveolar surface inside your lung. Now they will create fibers deposition. Now by utilizing this human alpha-1 antitrypsin, you can go for a recovery of that disease called emphysema. In 1997, Rosie, first transgenic cow, remember the Rosie, name of the first transgenic cow, produced human protein enriched milk. That is, human normal cow milk is not exactly similar to human milk. Cow milk has lactalbumin, but this human alpha lactalbumin is absent in normal cow milk. But what scientists have done, they have introduced human lactalbumin producing gene into the lactation cells of that Rosie cow, and the milk produced by Rosie is rich in human lactalbumin, that is human alpha lactalbumin, that used to produce that human protein 2.4 gram per liter. Okay, we'll also use transgenic animals for vaccine safety testing. Transgenic mice are used to test the safety of the polio vaccine. If it is reliable, they can replace the use of monkeys. So generally we use monkeys and chimpanzees because they have genetic similarities with us. Okay, first there will be lab trial, then there will be trial on animals, then there will be trial on monkeys and chimpanzees, then there will be trial on small population of human, then the successful trials, if there is full trials of certain vaccines, then they will be marketed for a normal population of a country, right? So instead of going for monkeys, utilizing monkeys and chimpanzees, because that needs very complex methodology to raise monkeys and chimpanzees into laboratories, and there are certain ethical issues, right? So rats and mice are easy model organisms for this vaccine safety testing. They obviously genetically modified rats and mice. Chemical safety testing, toxicity testing of some transgenic animals carry genes which make them more sensitive to toxic substances than non-transgenic animals. So they are exposed to the toxic substances and the effects is studied, it gives immediate results. Suppose you are coming with a new cosmetic, new cream, right, which you want to apply on skin, right? Before applying or before the trial on human being, you have to utilize genetically, if you utilize genetically modified organisms by introducing human gene, so it will be better studied and what is there any ill effect or any allergic reaction of that particular cosmetic component. These are some genetically modified animals or transgenic animals. This is genetically modified mice. So the fluorescent gene, in the beginning of the lecture I was talking about the glow gene, the fluorescent gene. There are two kind of luminescence in animals you can observe. One is phosphorescent, one is fluorescent. So what is difference between fluorescence and phosphorescence? So in darkness, if there is no light at all, the phosphorescent will start glowing. This is called bioluminescence. Okay, but what about fluorescence? You need a source of light, particularly UV light or blue light. So under blue light or violet or UV light, they will start glowing. So that is called phosphorescent. Basically, we take this GFP, green phosphorescent protein from certain species of jellyfish, and we introduce that gene into some animals like cats, monkeys, or even mice. So they will also exhibit phosphorescent under UV ray. So this is the first genetically modified monkey, we call it Andy. If you reverse it, it's DNA. Okay, this is the first genetically modified cow, Rosie. Okay, now we'll talk about, we have already spoken about different sectors where we can explore the real life applications of biotechnology. We'll talk about some ethical issues. Okay, so what do you mean by ethics? What's is everything should be binded with a limit. So you shouldn't cross a limit of anything, right? So that is called ethics. What we should do, what we should not do. Problems of unpredictable results, like for genetic modification may cause unpredictable results. Indian government has set up an organization which is called GEAC, Genetic Engineering Approval Committee, to make decisions about the validity of genetically modified research, GM research, and the safety of GM organism for public service. And few will say that this is against the nature, this is against the Almighty. So there are multiple issues are there like suppose you have produced a GM crop. You have produced a GM crop. Now you don't know upon human consumption that may exhibit toxicity, right? There may be some adverse effect. So in that case, the GEAC, Genetic Engineering Approval Committee, will give a ban on this particular GM crop. So that is the ethical practice. Okay. Now we'll talk about biopiracy. Now what do you mean by piracy? There is a term in the field of research, that is IPR, intellectual property right. IPR, intellectual property right. So there is a categorization that what are the things which will come under intellectual property, because everything we are researching, we are inventing, we are discovering due to our intellect, right? Now if you are whole and sole proprietor or responsible person behind a particular idea or a particular discovery, particular machine, particular technology, or particular crop, a new variety, then you should get the credit for that. But as because you don't have that much reach, you don't have that much uh, relationship or that much money power, some other company or some other influential person will steal that from you by giving you a lump sum amount of money. And by utilizing that particular discovery or particular idea, that company or that very influential person are gonna earn in crores, in billions. So that's why we need to talk about biopiracy. So this is the use of bioresources by multinational companies and other organization without proper authentication or authorization from the countries and people concerned. Now in daily life you can observe like examples of normal piracy. I'm not talking about biopiracy. Suppose you have composed a song, right? You have composed a song, you are the music director of that particular song. Now one big banner is coming with their upcoming film, and suddenly you observe that their film has used your song without giving you any share, right? Without giving you any money, without giving any notation, without giving you any like what do you say, without giving you any credit to that, right? So if a film is used utilizing your song without giving you any credit, then you can say that they are doing piracy against you. So obviously you can file a suit or legal suit against that. So one of the example is basmati rice has unique aroma and flavor. Okay. Now one very important thing you need to remember, that when you talk about India, from immemorial time we have nearby 2 lakh species of rice present, indigenous variety of rice we are cultivating in Indian fields by Indian farmers. Okay. So one of the varieties, basmati rice, is well known for its unique aroma and flavor. Now India has 27 varieties of basmati. Now what happened in 97, an American company called Rice Tea, that is the name of the company, got patent rights on basmati rice through the US Patent and Trademark Office. So that particular hybrid variety was originally from India. Now this allowed that company to sell a new variety of basmati. This was actually derived from Indian farmers' varieties. Now Indian basmati was crossed with a semi-dwarf varieties and claimed as a novelty. Novelty means their unique product. Okay. Now other people selling basmati rice could be restricted by patent. Now if suppose that's an American company, now they have taken that idea, that variety, everything they have collected from India, and they have, what they have done with that basmati, specific variety of basmati rice, they have crossbreeded a dwarf variety of rice and they produced a hybrid, and they said this is a unique one. Now they are selling into the market of US. They have taken their IPR on that, okay, legally. Now if you are Indian farmer and you have gone to USA, America to sell a species of another variety of basmati rice. Now as because in USA the patent is given on their name by the name of rice tea, then you have to share, you have to give them share whatever amount you are selling into US market. Okay, that is the drawback. Okay, that is actually biopiracy done by that rice tea. But Indian government also has some preventive laws against that. Few other examples are there. Herbal medicines like turmeric and neem are also examples. The patent should be for Indian companies or Indian government. Okay. Now Indian Parliament has cleared the second amendment of Indian Patent Law bill that has considered patent terms emergency provision and research and development initiative. Okay. Now few foreign companies, they were trying to take patent on these herbs like turmeric and neem, but as soon as Indian government got to know about this, so for all this product, now the sole and whole right or the patent goes for Indian government or Indian companies. Okay. So these are the things we have learned in this chapter. The green biotechnology or the agriculture based application. Okay. We talked about GM crops, we talked about improved crop yields, the selection of quality along with quantity, production of disease resistant crop plants, herbicide, pesticide resistant crop plants, stress tolerant crop plants. We have talked about biofortification. Okay, vitamin A and iron rich rice, that is golden rice. Okay, we have also studied the medicine, the application of biotechnology in medical field, recombinant therapeutic proteins. We have spoken about, learned about production of human insulin by utilizing E. coli bacteria, that Eli Lilly, humalin. Okay, and also few other things are there like monoclonal antibodies, tumor suppressor agents, interference, human growth hormones, recombinant vaccines are there, clotting factors are produced by utilizing rDNA technology. Okay, like when you talk about recombinant vaccines, second generation vaccine, that is hepatitis B vaccine, is produced by utilizing genetically modified yeast. DNA vaccines and RNA vaccines are also developed during the time of research, which was done to market some vaccines for COVID. Okay, and monoclonal antibodies which are produced by genetically modified mice by utilizing hybridoma technologies. We learn in higher classes treatment of genetic disorders, gene therapy. We have learnt about adenosine deaminase deficiency. Okay, we will learn in future days about environmental biotechnology, bioremediation. Okay, one of the great example for bioremediation is super bug. Basically, that's a species of Pseudomonas bacteria that helps us, genetically modified Pseudomonas or superbug. That's a bacteria that helps us to clear the oil spill on oceans by oil tankers. Okay, we use genetically modified microbes for cleaning of pollutants, right? We have learnt about transgenic animals and their utilization, and we also learnt about industrial biotechnology, how biotechnology helped us in large scale production of enzymes, hormones and other cosmetics, industrial products, biofuels, pharmaceuticals, and we have concluded the chapters with ethical and social issues. There you were talking about the patent bills, the IPR acts, intellectual property right acts. We have spoken about the 2 lakhs varieties of indigenous rice and how one variety of basmati rice was stolen by one American company, and they took the patent by doing merely easy hybridization with a dwarf, semi-dwarf variety of rice species, and they claimed as their own novelty one, as their own IPR, right? So we have spoken about, we have learnt about biopiracies. We have spoken about how Indians herbs like turmeric, neems, were claimed by few foreign companies, and then how Indian government protected those rights or patents of those particular things. Okay, so let's solve few questions, few important questions. So first question we are solving, we are going to solve now. There is how Bt toxin kill the target insect in protection of cotton plants. Midgut cell lysis and swelling, paralysis and loss of coordination, brain death, formation of abnormal proteins. Bt toxin, the protoxins get converted into toxin into the midgut of the larva of few insect, and they cause cell lysis and swelling and ultimately death. Next question, ELISA is based on A and used to detect B. Now A and B are respectively antigen and antibody. Antigen and antibody interaction. Antigen and antibody interaction. Okay. Now one assertion and reason question. Assertion said that RNAi involves silencing of a specific mRNA due to complementary RNA that binds to and prevents translation of mRNA. RNAi involves silencing of a specific mRNA due to complementary RNA. That is true. Now let's see what reason is saying. RNAi takes place in all prokaryotic organism. That is wrong. All eukaryotic organism. So that is false. So assertion is true, but reason is false here. Let's solve next question. An improved variety of transgenic basmati rice. Okay. Improved variety of transgenic basmati rice give high yield and is rich in vitamin A, golden rice. Next question, which is not a feature of genetically modified plants, made crops more tolerant to cold and heat. That is a feature. Reduce reliance on chemical relevance on chemical pesticide. That is also true for genetically modified plant. Cannot reduce post harvest loss. This is not relevant. Enhance nutritional value of food like golden rice. So our answer is option 3, which is not feature of GM plant. Cannot reduce, it reduces post harvest loss, we can talk about flavor saver tomato. Flavor saver tomato, remember that. Okay, so we are done with another chapter, that was biotechnology and its application. And again I'm repeating it, do not forget to check PW app, and obviously the new feature, that is PW NEET English in WhatsApp, and you have to just scan this particular QR, and that will open a door for you towards multiple things like questions, polls, handwritten notes, DPPs, practice sheets, mind maps, overviews of chapter, quick revisions, new strategies, and more and more. So please stay tuned. Thank you and bye-bye.